Now, types of protein. Those proteins which produce only amino acids on hydrolysis are called simple proteins. Their components are only amino acids such as albumin, glutamine, globulin, prolamine, histone, protamine, etc. Scleroproteins are also there, present in keratin, elastin, collagen, etc. And some proteins are conjugated proteins because of the presence of a non-proteinous prosthetic group. A combination of non-proteinous prosthetic group with such proteins make them conjugated proteins, such as chromoproteins, hemoglobin, rhodopsin. These are chromoproteins. They have colors. And this non-proteinous part, it is combined with non-proteinous parts and produces color. Then lipoprotein, combination of protein and lipid. It is present in egg and milk, etc. Phosphoproteins are also there, found in the in eggs. And nucleoprotein, proteins remain associated with DNA and RNA, and those are nucleoproteins. And while defining DNA, we say that DNA is a nucleohistone complex. Histone is a protein, or DNA is a nucleoprotein complex. Glycoprotein, protein in combination with carbohydrates, that is called glycoprotein. Yet another type of protein is found, that is derived protein. Actually, these are not present in nature, but during digestion or any other process of metabolism, some other processes of metabolism, they are formed, such as proteose, peptone, etc. On the basis of sources, proteins may be divided into two types: animal proteins, মাছ, মাংস, ডিম, এইসব যকৃৎ, বৃক্ক, এসবের মধ্যে থাকা প্রোটিন। অ্যান্ড প্ল্যান্ট প্রোটিনস, যেমন সয়াবিন, ডাল, এই বিভিন্ন রকমের কলাই, এমনকি রাইসের মধ্যেও উদ্ভিজ্জ প্রোটিন আছে। যে সমস্ত প্রোটিনের মধ্যে এসেন্সিয়াল অ্যামাইনো অ্যাসিডস সবগুলি রয়েছে আর কি উপযুক্ত মাত্রায়, সেগুলোকে আমরা ফার্স্ট ক্লাস প্রোটিন বলবো। সাধারণত প্রাণীজ প্রোটিনগুলো এই ফার্স্ট ক্লাস প্রোটিনের মধ্যে পড়ে। সেকেন্ড ক্লাস প্রোটিন সেগুলোকেই বলবো যাদের মধ্যে অত্যাবশ্যকীয় যে অ্যামাইনো অ্যাসিড, এসেন্সিয়াল অ্যামাইনো অ্যাসিডস সেগুলোর one or more such essential amino acids are absent in which proteins, সেগুলোকে আমরা বলবো সেকেন্ড ক্লাস প্রোটিনস, যেমন ডাল। অ্যান্ড in where the essential amino acids are absent and for which they cannot take part in nutrition or growth of the body of an organism. Such proteins are called third class proteins. Example, gelatin. There is no essential amino acid present in gelatin, and therefore it is it cannot take part in nutrition and growth of the body. So it is called third class protein. And then in the last part of our discussion, we will go to a different point, that is the structure of protein. We have seen that amino acids form peptide bonds, and many amino acids together form a polypeptide, and actually these are thread-like structures, and these form 3D structures by different methods. The primary structure is formed by the addition of amino acids one after another, and then the secondary structure is formed. That will be the point of our discussion now. So as a result of different type of interactions such as ionic bond, van der Waals attractions, hydrogen bond, etc., each type of protein assumes a three-dimensional structure which is determined by the order of the amino acids in its chain. The final folded structure is called conformation, and the protein folding has been studied in a test tube by using highly purified proteins. A protein can be unfolded or denatured by treatment with certain solvents which disrupt the non-covalent interactions holding the folded chain together. In a solution of urea, you know urea, CuNH2 twice, where there is one urea molecule per 6 water molecules in the solution. In such type of solution, if some proteins are mixed, they get unfolded, that is denatured. And look at this picture. Suppose this is the conformation of a protein. Amino acid, amino acid, amino acid, amino acid, etc. Polypeptide chain. These are peptide bonds. So we can assume it as a long thread. So replace it with this thread-like structure. This is made up of amino acids this is a actually polymer of amino acids, and this gets folded in such a manner, suppose in such a manner, it's the structure of a purified protein from cells, from isolated cells. Now when it is denatured, it will take this shape. This folding, this type of folding will disappear, folding will be lost, and this type of structure will be present. And when will it happen? I told you that in a solution of urea, where there are 6 water molecules and 1 urea in the solution, this can happen. And when this urea is removed, it refolds into this structure, that is, it renatures. It is called renaturation. আমরা এই ধরনের ডিনেচারেশন ডিএনএতে দেখেছি, হিট প্রয়োগ করলে তার যে হাইড্রোজেন বন্ডগুলো ভেঙে যায়, দিয়ে ডিএনএর যে ডাবল হেলিক্সটা তার দুটো স্ট্র্যান্ড সেপারেট হয়ে যায়, দ্যাট ইজ কল্ড ডিনেচারেশন। ডিনেচারেশন কেন? ন্যাচারাল কন্ডিশনে যেমন থাকে তার বিপরীত পরিস্থিতি, তাকে বলছি ডিনেচারেশন। এবং সেই ডিএনএর ক্ষেত্রেও আমরা দেখেছি, হিটটাকে যখন সরিয়ে ফেলা হয় তখন আবার ঠান্ডা হয়ে যায়, যে মিডিয়ামটায় রাখা হয় সেটা যখন ঠান্ডা হয়ে যায় তখন আবার ডিএনএ দুটো থ্রেড হাইড্রোজেন বন্ডের দ্বারা অ্যাট্রাক্টেড হয় এবং আবার ডাবল হেলিক্স ফর্ম করে। এই প্রোটিনের ক্ষেত্রটাও তাই, এটা ইউরিয়া ট্রিটমেন্ট করে এটা মানে ডিনেচার করা হয়েছে। ইউরিয়াটা রিমুভ করলে ইট উইল রিনেচার টু দিস কনফরমেশন ওয়ান্স অ্যাগেন। When the three-dimensional structures of many different protein molecules are compared, it becomes clear that although the overall conformation of each protein is unique, each protein's conformation is unique. Two regular folding patterns are often found in parts of them. There is somewhat similarity in the folding patterns. The first folding pattern to be discovered is called alpha helix. It is found in protein alpha keratin. Keratin is abundant in skin and its derivatives such as hair, nails, and horns. And a second folded structure is also found, that is called beta sheet. It is present in the protein fibroin, the major constituent of silk. Beta sheet. These two patterns are particularly common because they result from hydrogen bonding between NH and CO groups in the polypeptide backbone. Strictly speaking, this structure is like this, and this is the chain of amino acids, of which these are the polar side chains. These side chains are I want to remind you: alpha carbon H, COOH, NH2, and this is R. This is the side chain. The property of an amino acid depends on on this R part. So these are the polar side chains are of different kinds, polar, non-polar. You have known it. These structures are drawn as polar side chains. I have drawn a few just, and these are non-polar side chains. To differentiate them from the polar ones, I want to draw such type of diagrams, non-polar side chains. So while folding into this conformation, these parts will be there, and they will help in forming different type of forming different type of bonds. Again, I can show. I want to show it because the picture was not clear. Now I hope it has been clear to you. These are polar side chains. These means this part, and these are the non-polar side chains, means also this part, but there is no polarity found in this part. You know of non-covalent bonds. Covalent bonds are known to you when electrons are shared between two or more molecules, more atoms, and non-covalent bonds are also there, such as hydrogen bond, ionic bond, etc. So three types of non-covalent bonds have been discussed. One is ionic bond, another is van der Waals attractions between atoms, and the third one is hydrogen bonds, which is very important. Although a single of these bonds is quite weak, in case of DNA we have also seen it, that hydrogen bonds are quite weak. But given the enormity of the hydrogen bonds formed in a DNA thread, DNA double helix, the strength is very high. So many of these quite weak non-covalent bonds often form together to create a strong bonding arrangement, as in the example I am showing you, as in these three examples. But I will not draw such pictures because the video is getting lengthy. It takes time to draw a picture and the video gets lengthy. You just see these three pictures and three diagrams from your book. You see, this is the ionic bond. Between oxygen and nitrogen, here you see oxygen and nitrogen, and there is the ionic bond. It is negatively charged, it is positively charged, so there is the ionic bond. And this is the protein. This is one protein and this is another protein, and these are the side chains. There are ionic bonds. Also, you find in this picture van der Waals attractions here, and ionic bonds are present here, hydrogen bonds are present here. So in a protein molecule, more than one type of non-covalent bonds may be present. And also in this structure, you can see the hydrogen bond between between hydrogen and oxygen here, and this is the amino acid backbone. This is also, and these two remain together, which is necessary for folding and to keep the uh, amino acid backbone together. This type of hydrogen bonding is necessary. The pictures are obscured. Now let us draw one. Suppose this. And this, and a hydrogen bond is present. Here is oxygen, such and such. Here also, this is oxygen and this is hydrogen part, and carbon. This type of bond is present. It takes long time to make a diagram clearly visible to you. That's a problem of making video from home. Now it is visible to you, I think, clearly. You see, this is the structure of alpha helix, and this is the structure of at the bottom. You see the structure of another type of protein that is called beta sheet. Beta sheet মানে চাদরের মতো, and in detail in this picture, this one is alpha helix, and two alpha helices are coiled together. Three alpha helices may also be there, forming thread-like structures. The amino acid sequence of a protein is known as the primary structure of the protein. Stretches of polypeptide chain that form alpha helices and beta sheets constitute the protein's secondary structure. And the full three-dimensional organization of a polypeptide chain is sometimes referred to as the protein's tertiary structure. And if a particular protein molecule is formed as a complex of more than one polypeptide chain, such as in case of hemoglobin, you will see at a later stage that hemoglobin contains 4 polypeptide chains. In that case, such a protein, the complete structure of such a protein is designated as the quaternary structure. We have seen that protein molecules can have an elongated fibrous shape. Now we will see the beta sheet that we are talking about. Here you see in this picture, the protein globules are there forming a sheet, and these are hexagonal protein globules. They are placed in such a manner that it forms a sheet, SHEET, and this sheet is rolled into this pack, which is found in the capsid of a virus and in many other structures. This type of proteins are also found a single protein subunit assembly requiring multiple protein-protein contacts. Hexagonally packed globular protein subunits here forms a sheet, here forms a tube, and inside this tube remains the nucleic acid in case of virus. And specialized membrane proteins are sometimes arranged this way in lipid bilayers. With a slight change in the geometry of the individual subunits, a hexagonal sheet can be converted into a tube, you see, and with more changes into a hollow sphere, একটা গোলক ফাঁপা গোলকে পরিণত হতে পারে এরকম শীট দিয়ে. Protein tubes and spheres